Peace and blessings. Welcome back to another video. And in this series, we have been practicing some of the questions which have been asked in the previous examinations conducted by ICMR. And today, in the same continuity, we are going to discuss some of the questions which have been asked in the previous year examinations related to the DNA or related to the nucleic acid. So first, let's briefly rewind all of those concepts or the experiments which have been conducted related to the nucleic acid or the DNA. So it all started when a Swiss biochemist named Friedrich Mescher, he isolated a substance from the white blood cells and he named that substance nuclein. And then it was followed by the discovery by three different scientists, Avery, MacLeod, and McCarthy. In 1940s, they worked with a Streptococcus pneumonia. So it's a bacteria which is responsible for causing pneumonia. Now what they did, they performed an experiment in which they extracted the DNA from the virulent strain and then injected it into a non-virulent strain. They observed a phenomenon called a transformation, in which what they saw was that a non-virulent strain, it became virulent. That means that some factor was getting transformed from the virulent into the non-virulent. Later on, to confirm that, to settle down this DNA and the protein debate, in 1952, another experiment using the bacteriophages was conducted by Hershey and Chase. Then afterwards, we know that the model was proposed by the Watson and Crick. They gave the B form of the DNA. On the same line, in the late 1940s, Chargaff's rules were given by Irwin Chargaff and his colleagues. So what they discovered that in the two DNA strands there exists specific base pairing. He stated that the amount of adenine, it equals to the amount of thymine, and similarly the amount of guanine, it equals to the amount of cytosine. That means the purine is equal to the pyrimidines. And this finding, it hinted at the complementary nature of the DNA strands. So here we are not discussing these experiments, we are just going through that time scale. Now we saw the Chargaff's. Another important thing is not only the B form of DNA exists. So that was the model proposed by the Watson and Crick. Later on, it was also confirmed that there are certain other forms of the DNA which do exist, like we have the A form of DNA. And the only form of DNA so far, that is the Z form of DNA, it has a left-handed helical sense. So rest we see the B form and the A form, that is both of them, they have the right-handed helices. Another important thing from this, so from the exam point of view, this table is quite important. So another thing is with reference to the conformation of the ribose or the deoxyribose sugar. If you have a look at this structure here, the furanose ring of whether it is ribose or a deoxyribose pentose sugar, that is non-planar. Okay, so furanose ring, that means the ring which contains 5 carbons. And now, as I say that this is non-planar, so this non-planarity, that is termed as the puckering. And this structure can exist in two basic conformations. It can be either in the envelope form or it can exist in the twisted form. Now, what do I mean by this? In the envelope form, all of the 4 members, they are coplanar, and only one, that is the 5th, it will be away from the plane. Okay, but when I'm talking about the twisted form, 3 members, they are coplanar, and the 2 members, they will lie out from the plane. Okay, so in the envelope form, the 4 are coplanar, 1 is out of the plane, and in the twisted form, 3 are coplanar and the other 2, they lie away from the plane. So all of this is with reference to the furanose ring of the deoxyribose in the DNA. Okay, now in case of the envelope form of the deoxyribose sugar, this out of the plane atom, that is almost always C2 or C3, as you can see here. So it's always C2 or it's always C3. Now, such kind of a conformation, such kind of a puckered structure, which places either the C2 or C3 on the same side as that of the base, that is the C5. So we can say with relation to the C5, we call that as an endo. If the C2 position or C3 position that is on the same side of the plane as that of the fifth position. That means with reference to the base, then we call such a structure as endo. But if it is on the opposite side, you can see here, this is not on the same plane as that of the base C5, so we call this structure as C3XO. Remember that in most of the cases, when we talk about nucleic acids in the B form, A form, and Z form, mostly we have endo structures. It can be either C2 position or C3 position, but it is mostly endo. Okay, so these are some of the characteristic features of all the three different kinds of the different forms of the DNA molecule. Now, so this is the B form of DNA we have been discussing so far, and this structure was proposed by these two scientists, Watson and Crick. And the complementarity which we had discussed, this is you can see the first and foremost thing is that the DNA, it's an antiparallel structure. So if you see that one of the strand, it's from 5 prime to 3 prime, and another one, it shows an antiparallel orientation. Got it? So second point is that there is the complementarity. So second thing is that there is the hydrogen bonding between the bases. First thing, adenine, it base pairs with thymine via just two hydrogen bonds. So these blue dots, they represent blue dashed lines. These are the hydrogen bonds. Okay. Similarly, when you look at the G and C pair, G and C, they do the base pairing between them via three hydrogen bonds. So obviously, if here we have three hydrogen bonds, that means the more stronger one, that is the GC bond, than the AT bond. Okay. So all of this structure, it comes out using the Chargaff's base pairing tool, as we saw earlier. Okay. Now let's talk about some of the questions. Let's discuss some of the questions which have been asked previously. So this have been asked in ICMR 2023. It says that a piece of DNA was analyzed and 15% of its nucleotides, they were adenine. What percentage would be uracil? Now, usually if we look at the question, it is asking that a piece of DNA. First and foremost, DNA, it contains bases. Four bases are there. We have adenine, we have guanine, we have cytosine, and we have thymine. So there is no uracil. And if it's asking that what percentage would be uracil, it is 0%. That's it. Okay, so our answer here, it should be 0%, this one. Now, second, which one of the following, this was asked in ICMR 2021, it says that which one of the following forms the left-handed helix. So if you remember the earlier table which we saw, we should remember that the left-handed helix, that is only and only in case of the Z DNA, that's it. So in this Z DNA, you will see the left-handed. Rest, if it is B form of, you can see here from the table. If it is B form, it's A form, it has a helical sense, it has the right-handed helical cells. So here our answer, it should be only the Z form of DNA. Moving to the next, this question, it was asked in ICMR 2022. It says that if the amount of adenine and thymine in a double-stranded DNA is 45%, the amount of guanine in this DNA will be. So we have been given the amount of adenine and thymine. We have to find out the amount of guanine. Now, I have solved this question using the Chargaff rule. We know that the amount of purines is equal to the amount of pyrimidines. And if we combine all of the bases together, that is adenine plus thymine plus guanine plus cytosine, it gives us the complete composition, that is 100%. Now, what I have done is, so in the question, it's already saying that the adenine and thymine, that is the total amount of adenine and thymine, that is 45%. So we'll substitute in place of adenine and thymine 45%. Then afterwards, the amount of guanine, it's going to be equal to amount of cytosine. So in place of C, we can write down G. That's what I have given, 2G. Now solving this equation, it becomes twice G, it will be equal to 100 minus 45, and guanine, it will be equal to, it gives us 27.5%. So our answer here should be 27.5%, the second option. So all of the questions, they have been asked linked to the Chargaff's rule. Similarly, another question which have been asked, it is which one of the following is wrongly paired? So this is the question asked in ICMR 2010-22. Now, the question is quite simple. Question is that okay, so it's regarding the biomolecules and the kind of the linkages or the bonds which they have. Now, if we'll have a look, starting from proteins. Proteins, they are made up of different units. They are made up of amino acids. So the constituents of the protein are amino acids, and each amino acid is linked to one another by a bond called a peptide bond. Okay, so this is peptide bond. Okay, let's move ahead. When we talk about the polysaccharides, the polysaccharide, it's made up of it's constituted of different monosaccharides, and different monosaccharides, they are linked together by glycosidic bond. Okay, the second thing is done. And the third, that is nucleic acid. Now, if you look at the nucleic acids, we saw earlier, the nucleic acid, it's made up of different nucleotides which are linked together by the phosphodiester bond. So that means nucleic acid, that is a biomolecule, and its constituents are nucleotides. And nucleotides are linked by phosphodiester bonds, not the hydrogen bonds. So if it's saying that which is wrongly paired, our answer it should be first one, that is the nucleic acid and bonding hydrogen bond. It should have been nucleic acid. If it was linked with the phosphodiester bond, then it would have been correct answer. So that's the way we solve these questions. And in the next, we will discuss some of the another topic and also practice some of the questions which have been asked previously in the ICMR.